is a video for my honors biology students about photosynthesis, and this is part 1. All right, so I'm going to make myself a little bit smaller and present here. All right, so here we go. So the first thing we're going to be talking about is I want to remind you that the notes are down in the descriptors. If you would like to use those, the same ones that my students use, column 1 you can fill in and I'll help you fill those in, and column 2 add in images that are helpful for this presentation and for your review. All right, so first thing we want to talk about is the basics. So in photosynthesis, you're converting solar energy. You're converting solar energy into chemical energy. That's the goal, to convert solar energy into chemical energy of carbohydrates. And then the plants can use those carbohydrates they make for cellular respiration, or as animals, right, we can eat those plants and use it for cellular respiration. So autotrophs have the ability, on your notes, to produce their own food. Autotrophs have the ability to produce their own food. Whereas heterotrophs, right, let me get a pointer. Heterotrophs, you want to put on there, we are consumers. We are consumers. We take in preformed organic molecules and use those as building blocks and for energy, right? Okay, so we are consumers. So let's take a look at the reactants and products of photosynthesis. So this is the Minecraft version here. So take in your tree. I want you to take a look at where one of the items you need for photosynthesis is water. And so water's got to come in through the roots, and then it moves up through the tree, so it's available here in the leaves where they're doing photosynthesis. Now, what else do you need? You need carbon dioxide. That is going to be reduced into sugars. You need energy. This is an endergonic reaction, so you're going to need a little sunlight. Now, the output of that is you're going to have oxygen, which is also going to go right out of the leaves, and you're going to build sugars. Those sugars could be used for the growing parts of the leaves, or they could be transferred back down into the roots for storage. Now, I want you to take just a second and look at what else is going in through these roots. So you have different minerals that you need in order to build things, right? So you can see nitrogen. That would be really important for nucleic acids or amino acids and proteins. Phosphorus, right? ATP, right? The P is the phosphate, built out of phosphorus and oxygen. But look over here. Here's oxygen. Why does this tree need oxygen down there in its roots? Remember, who does cellular respiration? All cells. And so down here in the roots, they're going to be doing, and throughout the entire plant, they're going to be doing cellular respiration. So oxygen gas is needed right here in the roots. So on your notes, let's look at reactants. You need solar energy, that's one reactant, and water and CO2. The water, on your notes, in through the roots. The water is in through the roots. The CO2 is going to come in through openings in the leaves. I'll show you a cross-section of a leaf in just a minute, called stomata. So in through the stomatal openings in the leaves. And then both water and CO2 are going to diffuse into the chloroplast so it can do photosynthesis. So let's look right here at a cross-section of a leaf. This right down here at the bottom, that is the stomata. Those are openings in the leaf for gas exchange. And then when you look at individual plant cells, you can see those dark green little disc-y looking things. Those would be chloroplasts. Remember, they have a double membrane. And then when you go to the interior of the chloroplast, you can see these stacks. These stacks are called thylakoid membranes, and the space around it is called the stroma. Let me show you another picture of that. Okay, so here you can see the chloroplast. You can see there's an outer membrane and an inner membrane. These right here, these stacks that you see, that's where the light reactions are going to take place. And the pigments are embedded in those membranes that can capture the light energy. And remember, the first law of thermodynamics says energy cannot be created or destroyed. It can just change forms. So what you're doing is you're changing light energy into chemical energy. And that chemical energy you're going to make in the light reaction is both ATP and reduced NADP. Then in your light independent reaction is going to take place here in the stroma, the space around these green hollow pancakes. And this is where you're going to use ATP and reduced NADP from the light reaction to reduce CO2 into sugars. All right. So on your chloroplast anatomy, it's a double membrane. The stroma is this fluid, semi-fluid interior, and then the thylakoid membranes. And if you notice all the folding, what's that folding going to do? It's going to increase the surface area for those reactions. So for increased surface area, pigments are embedded in those membranes. All right, good job. Let's keep going here. So this is all contingent upon light, right? So let's analyze light a little bit because that is our source of energy for this endergonic reaction. So this is what's called the electromagnetic spectrum, okay? And if you look across here, you're going from the tiniest of waves here on the left to larger waves like AM radio waves and FM radio waves. And right here in the middle is kind of blown up for you here is the visible light that we can use in photosynthesis. Not we, but plants, okay? So the smallest wavelengths down here, looking at 400 nanometers, and then the wavelengths get bigger and bigger and bigger here to red. Now, one thing I want you to get in your head, the smaller the wavelength, the more compact, the more energy. The bigger the wavelength, there's less energy. That's why an X-ray, right, has more energy in it than a AM radio wave, all right? So next, there we go. Let's talk about what happens with light when it hits something, okay? It can be reflected. For instance, I purposely wore a green shirt today. I don't know if you can tell that it's green, but there are wavelengths coming in from my window in front of me and the lights above me, and they're hitting my shirt, and then they're bouncing back off to you. It's getting reflected. That means I'm not absorbing the green light. Okay, or there, I have green earrings on here. It's reflecting green light back out to you in your eyes. Other colors like blue and red, those are getting absorbed, right, by the shirt because you're not seeing those colors, so I must be absorbing it. And then if it's thin enough, light could pass right through it and it could be transmitted as well. Now, if somebody was wearing a black shirt, that means they are absorbing every single wavelength there is. If you have a white shirt, it means you're reflecting all those wavelengths of light. So on your notes for pigments and photosynthesis, light is either absorbed, transmitted, or reflected. And so you have what's called an absorption spectrum when you look, oh, one more picture here. So here comes light, and you know, like my shirt, right? Most plants appear green, so they must be absorbing those other wavelengths of light and reflecting back out that green light. And sure, some light is actually getting transmitted and moving right through it. So light is either absorbed, transmitted, or reflected. Your absorption spectrum, okay, if we look here, we're going as we move from left to right, the wavelengths get bigger, and the biggest energy is the smaller wavelength. So here is our visible light. So you've heard of Roy G. Biv, right? As to remember the colors, red, orange, yellow, Roy, blue, indigo, violet, as you move across the spectrum, with violet being the most powerful. But on your visible spectrum, notice green right here. Most plants appear green, so they must not be using this light, but because your plants don't appear purple or red, they are using those two wavelengths. So let's look at some of the different pigments that plants have. Now, the most predominant pigment they have is chlorophyll, and there are different types of chlorophyll. Chlorophyll A can absorb, right? It can take in these purples and these blues, and then here on the other end, these reds and some of these oranges. Chlorophyll B, just to the interior of that, okay, can more of these blues, and then here on these oranges. Carotenoids, what do you hear in that? Carrots, right? So what color are carrots? Orange. And that's because they cannot absorb the orange wavelengths. You can see they're getting just down here in the purples and the blues and the greens, and so they reflect back the orange and red, and that's why it appears that color. So when you sum it all up for a plant, you look at its action spectrum or its absorption spectrum. And so it can really absorb on the visible spectrum, mostly on either end. So on your absorption spectrum, it is a graph of the percent of light absorbed at each wavelength by each pigment, A and B. Now, where do they absorb this? They absorb these light in, one other thing, yes, as you approach fall, the leaves don't appear green, right? Chlorophyll is the most abundant pigment, so it overwhelms all the other pigments like the carotenoids. in a plant as long as it's there and active, but it is not very stable and it easily breaks down in the colder weather. And you see some of these other pigments that can absorb other wavelengths of light. All right, so here, okay, let's talk about what these pigments do. I know this is a little complicated, but you'll get it, I promise. Take a look right here. When I talk about photosystems, and we'll get into the details a little bit later, remember when we looked at those thylakoid membranes, those green pancakes inside the chloroplast? Embedded within those membranes are photosystems. Here's photosystem 2, here's photosystem 1. And what is inside of these photosystems are various pigments that can capture that light energy and become energized by it. The electrons within them get excited, and then they move through a series of acceptors within the membrane of the thylakoid. And then they can pass these electrons along in an electron transport chain. So photosystems are where it all starts. You have pigment complexes, which are several pigments all together. And these are electron acceptor molecules embedded along this membrane. So the pigments get excited, their electrons within those pigments get excited, and they pass them to a series of electron acceptor molecules within the thylakoid membrane. So let's look at the overview equation of photosynthesis, and we learned this before, and you have the equation already written in your notes. So you have energy from the sun, and we're going to reduce CO2 into glucose, and we're going to take water and break it right at the beginning of photosynthesis and release oxygen. These hydrogens that you see right here that are part of water are going to be transferred temporarily to our energy transfer molecule, our mule, NADP, and NADP will carry those to the light-independent reaction and use the hydrogens from water to reduce CO2 into glucose. And you have that whole equation right there. So when it says the role of NADPH, it temporarily will hold the electrons from water and the hydrogens from water, and they are ultimately going to be given to CO2. So he's our mule to carry these hydrogens and their accompanying electrons over to CO2 in order to form glucose. Now, there are two overall reactions, the light reaction or light dependent reaction and the light independent reaction. The light independent reaction, like when I went to school, was called the dark reaction. And that's a little bit misleading because then you would think that it has to happen in the dark. And so that's why they call it the light independent reaction, that you don't need light for this part of it. And then Calvin, that's one of the guys that discovered the steps of it. So that's the other name for it. Okay, so old school dark reaction, Calvin cycle, or light independent reaction. The light reactions themselves occur on those thylakoid membranes, those hollow pancakes, and you can see water. You need water because you need a source of electrons, and immediately the water is going to be broken in half. So this O right here in the water is this oxygen right here. These hydrogens ultimately are going to be given to NADP to form reduced NADP, and it will carry it here to the Calvin cycle so that it can be given to CO2 in order to form sugar. All of that is going to require energy in order to build, because remember we talked about catabolic breaking, anabolic building. So we're building sugar, that's going to require ATP. So the light reactions also generate ATP for us to use in the Calvin cycle. When you're done with the Calvin cycle, then this mule has given up his hydrogens, he's oxidized and can be reused again. And the ATP will have been broken apart, releasing their energy and transferring that energy to building sugar so they can be reused again as well. So the two reactions, you have the light dependent reaction, water is split, light energy is used, and NADPH and ATP and oxygen are formed. And then the Calvin cycle, the light independent reaction or the dark reaction, you will use the ATP and the reduced NADPH from the light dependent reactions to reduce CO2 into sugar, into glucose, into carbohydrates, right? Okay, so let's follow just the light reaction for this video. So look at your big picture here. So here comes light energy coming in, water's coming in, it's going to be split, and oxygen will be released. And our goal here in the light reaction is to form ATP and reduced NADP that can be used in the Calvin cycle out in the stroma. So here's how it happens. So those two photosystems that I talked to you about, and I'll kind of build up on this a little bit. So this, keep in mind, where this is occurring is in the membranes of those thylakoids. Let me go right back here. See these thylakoid membranes, these green hollow pancakes? That's where this is occurring. So you have photosystem 2, and it was discovered second, so that's why you see it right here. Water is going to donate some electrons in photosystem 2, and there are pigments right here to absorb this light energy. And these electrons are going to get excited by this light energy and jump to a higher energy level. So now these electrons, through a series of redox reactions, can be passed to some acceptors here who will accept those electrons. And in the process of photosystem 2, you will ultimately concentrate hydrogen ions inside the interior of the thylakoid membranes. And just like we made a concentration gradient with hydrogen ions in the mitochondria, right? And we can use that, harness that to make ATP. So by concentrating those hydrogen ions in, when they go back out through ATP synthase complex, you can make ATP. And that's the long and the short of it for photosystem 2. It's all about generating ATP. Then these same electrons that went through photosystem 2's electron transport chain get excited again. They get to go on another ride, and this time another series of redox reactions, but you're not going to let them go all the way down to a low energy level. You're going to give them to NADP ox, forming reduced NADP. Now, this is called non-cyclic because electrons are coming in via water, going on a couple rides, and going out via reduced NADP. Let me show you another picture of that. Okay, same idea, just a different picture. So here you have water split. Here you have all your pigments to absorb light energy, and we take the electrons from water and get them excited, and they go through a series of electron transport chains, and and along the way there's a molecule called PQ who's actually bringing the hydrogens to the interior of the thylakoid membrane, and you can use that to make ATP. Again, the electrons get excited, go through another electron transport chain, and you use that to reduce NADP. So on your notes where it says electron flow in light reaction non-cyclic, photosystem 2 was discovered second, so we're starting right here. Water is split, forming oxygen and hydrogen. Water is split, forming oxygen and hydrogen. The hydrogen donates its electrons and then becomes concentrated inside the thylakoid membrane. The electrons are excited by solar energy. These excited electrons move down an electron transport chain. Here they are. The energy is captured by PQ. That's a molecule in here. I'll show you in just a second, who brings additional hydrogen ions. So not only the hydrogen ions from water inside those pancakes, but PQ brings hydrogen ions from one side of the membrane to the other. As the electrons move through the electron transport chain. And then we pass those electrons on to photosystem 1. In photosystem 1, the electrons are excited a second time. The electrons are excited a second time by solar energy. And ultimately, those electrons are given to NADP. Okay? And then this will be used by the Calvin cycle, your dark reaction. So let's take a look again. Okay, so here's photosystem 2. Look right here, water is split. Okay, these electrons are excited and they through the electron transport chain. This is all part of the first electron transport chain. Look right here, this is PQ. PQ is bringing hydrogen ions from the stroma to the interior of the thylakoid lumen inside the hollow pancakes. So now we have those hydrogen ions that PQ brought in, but we also have the hydrogen ions from splitting water. So those hydrogen ions can then cross back out into the stroma through ATP synthase complex, which you can use to make ATP. Okay, so that's photosystem 2. The same electrons in photosystem 2 are excited again, but this time we use those excited electrons to reduce NADP. So now we have out in the stroma reduced NADP and ATP, which can be taken to the dark reaction, the Calvin cycle light independent reaction. So electron flow in the light reaction. Oh, let me keep going here. Let me go right here. Skip. No, actually, so sorry. This is called, remember, non-cyclic photophosphorylation because electrons are coming in via water and then going out via reduced NADP. So cyclic, contrast this to cyclic photophosphorylation, which is not as efficient, okay? Because what happens in cyclic is the electrons just go roundy round. So the electrons get excited, okay? And they na-na-na-na-na-na and create a hydrogen ion gradient, but all they make is ATP. And so this is used as backup because you don't make any reduced NADP. I would think of this like you would think of anaerobic respiration fermentation, which is used for backup for aerobic respiration, and it's just not as efficient. So on electron flow in the light reaction, cyclic, not as efficient. As non-cyclic, water is not split and only ATP is formed. So let's look at that ATP formation. So this is a picture from your book. Same thing, just another picture. So here's photosystem 2. Water is getting split, right? The electrons are getting excited to a higher energy level. They're moving through photosystem 2, na-na-na-na-na-na-na-na. Here's PQ embedded in here, bringing hydrogen ions from the stroma into the interior of the lumen of the thylakoid membranes, creating this hydrogen ion gradient from both the water and from what PQ is bringing in. And this, when they go out through ATP synthase complex, you can use that to make ATP. Okay, so the electrons that went through photosystem 2 now go through photosystem 1. They get excited again, but this time you use that high energy to reduce NADP. So here is another picture of that. So you can see it one more time. Photosystem 2 and photosystem 1. So on your ATP production, the interior of the thylakoid membrane serves as a reservoir for hydrogen ions coming from the splitting of water, which you can see here, and from PQ. And this generates an electrochemical gradient, a difference in concentration, charge, and pH. So just like before, so they want to go back out. And so hydrogen ions move from the higher concentration inside here, the thylakoid lumen, back out to the stroma using ATP synthase complex channel. As the hydrogen ions use the ATP synthase complex channel, ATP is formed. All right, so that is the light reaction, and in video 2, I will teach you the Calvin cycle where we're going to harvest this energy from ATP and reduce NADP in order to make some sugars. Okay, great job.